Okay, good afternoon, everybody. Thank you for joining us for this session. I hope you all had a good lunch, and we won't allow you to settle too much. It'll be an energetic performance with this cast. Today we're here to discuss broadly the unique risks and benefits posed by novel therapies for age-related disease. We're really going to split this into two dimensions. One, I think, are the systems level risks, so to do with the R&D process and reimbursement, how we're valuing these therapies. And then the second component is very much looking at the product level risks, how they're used and how they're regulated. Joining me on the STAR study panel is Richard Barker, Director of CASME and the ex-head of the ABPI, which is the UK equivalent of our pharma, and a huge advocate for novel regulatory processes. Chris Hornsby from RMS, who you may have heard yesterday from his colleague, Peter Nikita, developed novel healthcare risk management tools. Brock Reeve, Executive Director of the Harvard Stem Cell Institute. He's spent the last few years corralling scientists in a useful direction. And finally, Anant Jani from Better Value Healthcare, which I think are dealing with a lot of the issues that we face today, which are what is the cost of a cure and how can we ensure maximum patient access. So even though the panel's meant to go until 3:30, we all have lives and risk benefit can't be spoken about for that long. So after briefly setting the scene, we're going to try and wrap up by about 2:30, 2:45. I've got a few slides to kick off with, I think, but I have no idea where the pointer is. Thank you. Okay. So first and foremost, as I mentioned, we have really the system level risks. Mike West yesterday spoke about the tidal wave of age-related disease. I think that's absolutely true. But what's going to solve that? It's going to be the individual products. And they're very heterogeneous, as you can see from the bag of Skittles we've got there. Some are therapeutics, some are devices, some are surgical interventions, and others are ancillary technologies which support their adoption. So looking at system risk, this is the classic translation pathway from lab to bedside. And we can see that there's a number of protagonists, academia and industry, and the regulator is the gatekeeper at all points. So we have unique risks related to R&D. So whether it's in-house within the companies or within academic institutes themselves. Yesterday we learned about novel academic industry partnerships and how they de-risk the translation pathway. No doubt with Brock we'll hear about more of those today. Secondly, regulation and particularly advanced regulatory, novel regulatory models. Adaptive licensing, one of Richard's passions, I'm certain will crop up, and also the FDA breakthrough status. Early this morning, there was a session in one of the parallel tracks looking at cancer therapies, and as many of you will know, the CAR-T work from UPenn was one of the first drugs to get the FDA breakthrough status. So there are regenerative medicines using these novel regulatory pathways as we speak. Finally, reimbursement. It has to be said that a lot of the regenerative therapies which we see today haven't been fantastic at being reimbursed. A, because reimbursement has been an afterthought. B, the efficacy versus standard of care hasn't been that clear. But also, I don't think people have necessarily wrapped their head around what value each stakeholder needs and gets, direct versus indirect benefit. And that's certainly something Anant will talk about later. So we've looked at system risks very broadly, but what about the product risks individually? And how can we stratify it? So firstly, patients. What are their existing comorbidities? What are their existing treatment regimes? All these things are very much going to take one homogeneous product and give it heterogeneous potential outcomes and introduces uncertainty into this risk-benefit matrix. Secondly, the clinical indication. For most of the age-related diseases, are we actually going to have transient therapies, which we take, they elicit a pharmacological effect, and then leave the body, or we're going to have single-dose cures? These elixirs may sound wonderful, but how do we regulate them? How do we objectively and systematically assess their risks and benefits? Secondly, the intervention, we mentioned there's a heterogeneous group of technologies. The way in which we evaluate the risks and benefits of devices from a regulatory viewpoint is fundamentally different to how we regulate therapeutics. So when we look at things like tissue-engineered scaffolds, which James Yu was talking about yesterday, they're combinational therapies. From a regulatory viewpoint, they share the same challenges as devices and the therapeutics, but leveraged together. And finally, the clinician. There's a number of clinicians at this event, and particularly in this room, and I know that we've all been talking about expanded access. And actually, the clinician has a really important role here. The named patient exemption, humanitarian device exemptions, are critical for allowing patients access to these somewhat experimental therapies early. But also, looking at the interface between system risks and product risks. Is access by one patient to one product enough for the entire system to benefit later? So this is simply a slide which I thought would be interesting from Chris's viewpoint and Peter's yesterday. In finance, we manage risk, benefit, and uncertainty relatively clearly. The risk is the risk of a loss. The benefit is a gain. I mean, uncertainty is really very much the unexpected. Is it a standard event where the covariance hold, or is it an outlier, a catastrophic event like the financial crisis? RMS have worked all that out, so we're going to be fine. But in healthcare, what's the analogy? So the risk is an adverse event, whether that's the worst outcome, death, or some unwanted inflammation. The benefit is obviously efficacy to the patient. But the uncertainty is totally unpredictable, because actually it's not just as simple as does it cure or not. It's arbitrary. It's a patient-reported outcome. It's a spectrum of the patient can totally climb the stairs and sprint up them versus they couldn't walk at all and now they can climb them slowly. It's much harder to assess that. Referring back to the challenge of combinational therapies, I always think that it's interesting to look at, well, fundamentally, the polymers which we've used in household objects, from clothing to mattress stuffing to underwear, never went through a regulatory process. But when we start using them in medical devices, all of a sudden, they have this new status. And the risks and benefits are elevated, or at least in many cases, poorly understood. So the crux of this, and I guess the inspiration for the session, and certainly the work that SENS funds focus on this, is both the FDA and the EMA in recent years have requested a much greater focus on risk-benefit analysis. They've chosen to call it benefit-risk, I think, for PR reasons. But either way, how can we more systematically and transparently assess risk and benefits of potential products and engage a greater number of stakeholders along the way? The challenge here is multifactorial, but the two key things are, one is it needs to be a decision support tool, not a decision-making tool. And the second thing is that, actually, how can you produce a public document which is accessible and accurate to all stakeholders, but that also maintains its core purpose, which is to evaluate a relatively complex product. So if we take what a patient wants to see and can understand, it's very different to what a regulator wants to see and can understand. So there's going to be different levels of granularity in any report. And as you can see, the stakeholders, using the wonderful CASMI logo, are incredibly heterogeneous, but they all form this systems challenge. Something which hasn't been spoken about much at this event, but I thought from the program it would be, was combinational therapies, but using a combinational diagnostic. So Herceptin and Oncotype DX is obviously a common example. But to what extent does a combinational diagnostic actually decrease system risk in the development pathway for a drug? And to what extent does it actually increase the risk or the uncertainty? So you can see here there's a vicious cycle between actually, you may get a lot more data, but A, how do you do it? You're going to find something you don't necessarily want to see, and also the development costs all of a sudden increasing. Alternatively, it may simply be that in the virtuous cycle presented here, you start out with a very clear end in mind, and it truly does reduce uncertainty and enables expanded patient access. Or, again, to revert to the talk yesterday, the Cystic Fibrosis Foundation, Calico, the cystic fibrosis drug, it's got a combination of diagnostics, so the right 2% of patients get it. Does that decrease risk to the overall system? Well, perhaps because 98% of the patients aren't using a drug which costs a lot and won't work, but then how does that impact the cost of it for the 2%? So thank you to all the funders as always, and now it's my pleasure to introduce Richard Barker from CASME, who's going to give us an overview of all things regulatory, I expect. Thank you.
David gave me a rather mouthful of balance and rigor and adaptability and so I'm not sure about that. But I thought I ought to at least make nodding acquaintance to it by putting it on the title slide. What I would like to do is to reflect on where science is taking us, but even more to reflect on the paradigm or mindset shifts that we are in the process of making on many dimensions, actually, including the dimension of adaptive licensing, adaptive development. So this is us, but you've probably heard of us already, so I can skip that. Because the new technology that we're talking about at this and many other meetings are opening up dramatic new possibilities. But we are facing an innovation process that I would argue requires major overhaul and is beginning to get it. This is not that the sky is falling. We're beginning to see greater flexibility from regulators and others. But I would like to call out 7 major mindset shifts that we need to turn, we need to make those shifts and turn those into practical action. So I probably haven't got all the right technologies here, but genomics, faster, cheaper, more insightful. Epigenetics, which I like to think of as personalizing our own genome. Multi-omics, some very interesting papers of late, including a very interesting paper from Michael Snyder from Stanford, where he basically sort of did a molecular X-ray of his whole biology as he went through a series of health events. He basically did everything. He did the genomics, the gene expression, proteomics, metabolomics, immunophenotyping, and basically figured out what was going on in this one individual as he went through some health events. And I think we'll see more and more of that, and that will open up all kinds of opportunities to do the kind of individualization and personalization that David was talking about. We're learning more about the biological networks in systems biology, and imaging is going to greater and greater levels of definition, even to the point of being able to track cell groups as they migrate around the body, and other things too. And here's just the example that you'll all be aware of, that we're beating Moore's law now with regards to the pace and cost of gene sequencing. And we are beginning to see the genomic markers feeding their way through into therapy choice. Again, David talked about this, which is the combination or companion diagnostic therapeutic approach. We will see more of these, but actually we will go further because full genome, next generation genome sequencing will enable us to completely individualize our perception of every individual tumor and maybe even colonies within the same tumor, and things that appeared to be similar to the clinician in the past or even the pathologist in the past will actually be teased out to be completely different and requiring a different cocktail of either chemotherapeutic drugs or CAR T cells or whatever in order to hit those very specific mutations. And I picked this chart up from a publication on mutations that are underlying that. I have no idea what the chart means, but I think it's one of the most beautiful charts I've seen. But you will have read some of the work, particularly in lung cancer, where we're not just doing a snapshot of what's happening in a tumor, we're actually tracking the evolution of the tumor and the proliferation of mutations. And so in terms of genomics, we're doing some fantastic things in terms of proteomics, in terms of metabolomics, all of these things. But the reason that I and others founded CASME is because I believe that there is this fundamental gap between the pace and breadth of innovation that's happening in the bench, happening in basic bioscience, and what's coming out in terms of fundamental patient benefit. I don't just mean new product ideas. I mean actual transformational therapies being applied to patients, and they're getting the benefit. Of course, the bottom curve is rising, but actually I would argue that the gap between what we think we might be able to do and what it turns out we can do is actually increasing, or has been until recent years. And so I think about this as addressing at least 4 gaps in translation. You'll be familiar with them. The gap between basic bioscience and having innovative products in the process of clinical trial, which a lot of people call translational medicine. Then the gap between having those products in the pipeline and actually having them regulatory approved and reimbursed. So the productivity, so to speak, of clinical development and regulation. A third area, gap 3, which I didn't used to think about much until I had a colleague called Rob Horn from UCL, who's a behavioral health person. And we're doing some more work at CASME thinking about how you can ensure active diffusion of new technologies once approved and reimbursed. And even more importantly, what is it about patient psychology that means even if you've got a great drug or a great device or a great therapy, patients don't adhere to it? What are the useful individual factors? Indeed, I would argue that personalized medicine has at least 2 dimensions, the biological dimension and the behavioral dimension, and you need to understand both of those in order to get personalization to work. And then the fourth one, which people more and more are talking about, which is feeding back from this big data that we're supposedly having. It's actually a big pile of garbage most of the time, but we are going to be able to get all of that into some sort of order. And what are the feedback mechanisms from how patients actually react to actual therapeutic regimens to what does the R&D process need to focus on in the future? But I think more fundamentally, more philosophically, maybe more suitable for just after lunch, are these 7 enabling mindset shifts. The first is people talk a lot about personalized or stratified medicine. Actually, I think that's a transition state between the definition of disease by symptoms and the part of the body in which they occur to a genuine molecular taxonomy of disease. And one of the things I applaud about the work that SENS is doing is forcing us to look down in the pyramid to say what's basically happening to the biology of this person that may show this symptom in one person and another set of symptoms in another. In other words, be able to drill down to what's actually happening at the molecular level and define the disease in terms of those molecular events. The second is, I think, an increasingly important network-oriented philosophy of discovery. I'm going to talk a bit more about each of these, so I should avoid the temptation to expand on this. Open innovation versus closed and heavily patented, protected. Collaborative approaches to evidence generation evaluation. Adaptive development and licensing, which David threatened I will talk about briefly. The process of patient engagement in the whole R&D endeavor. And systematic real-world outcome tracking, which is, I guess, my gap 4. So I think that we have had a rather simplistic and apparently rather unsuccessful view of drug discovery in the last 20 years. Basically, we have sought single disease-specific intervention points. We want to know what gene is associated with the disease, what protein does that code for, can we find something that binds tightly to that protein, and can we turn that probe molecule into something that looks like a drug. I think increasingly we're realizing that biology is a self-correcting network. Many diseases share common mechanisms. We've already talked about some of those examples today. And the goal is to modulate these. And I don't know if you agree with me, and you'll tell me if you don't, in a sense, using the homeostatic processes within human biology and helping them correct themselves rather than trying to defeat them. And that's a different way, I would argue, than the way most of drug discovery has worked in the pharmaceutical industry in recent years. This molecular taxonomy of disease I already talked about, but thinking about things at the molecular level and drilling down to that level. Open innovation, at CASME we've done some work recently on how open innovation is operating in the life sciences arena, because we know we've moved from a relatively patent-dominated discovery system where the concept of a patent is rather like the concept of land in the US legal system. It's an intent to exclude people from a space. And that's the role that the patent has had. I'm sure there will be people who will argue a little bit about this. As opposed to, for example, water rights and the ability to be able to share intellectual property as a field evolves. So this open innovation mindset that shares that intellectual property more broadly at the stage in which we really don't know what it'll lead to, and of course the vast majority of things that are patented in life sciences never produce products, to a system that we have yet to design, but I would call for, which is one that protects the major investments that come later in clinical development by granting various forms of market or data exclusivity to the companies who make those investments. And I think we would see a leap forward. We are beginning to see a leap forward through collaborative open innovation efforts. If companies share things, if companies and other parts of the healthcare sector share things early on, as long as we can solve the subsequent problem, which is nobody will invest in clinical trials and the hundreds of millions of dollars to do that unless they can be assured of return. Here is, I think, the biggest philosophical challenge the life sciences world faces. If you think about the regulatory and reimbursement process, the whole concept behind this is that you've got untrustworthy innovators who seek to get their products through the regulators and the reimbursement agencies who are watchdogs on behalf of society. That is the mental model we've lived with, I think, probably since thalidomide, actually, and maybe even before then. Increasingly, in Europe, we're seeing some very interesting experiments in this regard. Can we get multiple stakeholders together to actually collaboratively design the evidence generation package and how that evidence is going to be collected early in the process, maybe even before phase 2 if we're talking about a drug, so that both the regulators and the reimbursement agencies say, look, if you can prove this, you're on a winner. If you can't, let's talk about it. And the company knows, in a sense, in advance. Now, of course, we have steps in that direction. People like Bob Clay in the audience here has been part of scientific advice processes. We've had scientific advice, but then we had this fascinating debate recently in the literature about people saying regulatory authorities shouldn't even give scientific advice. That's sort of helping the enemy. So this mindset shift, which is that we should bring together early in the development process, not just the innovator and the regulator, but the innovator, the regulator, HTA agencies, payers, and the patients to design that evidence package. And that is part of the new EU adaptive licensing approach. And the other part of it is this: instead of insistence on a definitive one-time verdict on a potential new treatment, the leading light here is a guy called Hans-Georg Eichler at the EMA, and he talks about the magic moment where it goes from being no to being yes. Instead of that, we need the recognition that a promising therapy has to go through several stages of experimentation in a steadily increasing population so that we really understand not just what it would do in the perfection of a clinical trial, but what it does with the real-world patients with all their comorbidities. And there will still be, harking back to David's point, a lot of uncertainty at many stages here. We need to learn to embrace uncertainty rather than ignore its existence. And, whoops, I don't know what happened there. Can you get me back 2 slides? Anyway, the last point was fundamentally about patient engagement. Patients have been, shall I go forward from there? That's probably the thing to do. Patients have been the passive recipients of the benefit of innovation for too long. What they need to do is to be involved not just in this design process, but even involved in the definition of what benefit looks like and what people should be working on. Because as we saw in the example of cystic fibrosis, which again David mentioned, you had Bob Beal and the Cystic Fibrosis Foundation involved at every stage in the process of conceptualizing and taking that product to market. And then finally, systematic real-world outcome tracking from what I call a fire and forget clinical philosophy. Unless the person turns back up in the doctor's office, we treat every therapy decision as a chance to learn about that therapy as well as about that patient. So what I'd throw out there as a question is, where do we stand on making these? If you go with me at least part of the way that these are mindset shifts we need, where are we on those mindset shifts? At a totally sober lunch. On molecular taxonomy, I think it's spotty. We were getting there in cancer. We're getting there in some rare disorders. I think we're emerging a sense of how you need to work the network, not just the individual protein target. I think there are lots of very interesting experiments on open innovation, but I don't think there's a thesis out there on how it should work. Collaborative evidence and generation evaluation, I've already pointed out, is pretty controversial certainly amongst activists. And we now have a real step forward, I think, in Europe, and we've been part of calling for adaptive licensing, so that's now being piloted. I would still argue that the patient's engagement in this is rare, and we need to completely rethink how patient organizations should be involved. And then I think we need to take systematic real-world outcome tracking from all the talks and all the conferences on big health data to some useful action. So I think that's all I have to say. Thanks.
Thank you, Richard. So now it's a pleasure to introduce Chris Hornsby from RMS, who's going to tell us about how you're actually going to operationalise these models. And I've seen the slides already, and they're quite entertaining, so you should have some fun.
They're not supposed to be, but thanks for that. So RMS is a firm in the financial services and insurance industry, and over the last, let's say, 10 years, we've actually seen a fairly big change in the way that insurers and banks are regulated. And there is a large institutionalized and active body of legislation that's purely based around quantitative modeling. So the way an insurance firm operates and the degree to which it can sell business based on how much cash it has in the bank, this is all determined by quantitative models. And if you read a regulatory document about the do's and don'ts for an insurance firm, they're couched in mathematical language. So the fundamental essence of insurance regulation is, over the next year of operation of an insurance firm, the probability of insolvency has to be less than 0.5%. And we have the value at risk measure that came out of the banking sector and JP Morgan and so forth. So there is a precedent for ratified quantitative regulation. And this presentation is just some very sort of gut reaction thoughts to the topic about what that possibly could look like in regards to the way novel therapeutics are regulated. And it's designed to be thought-provoking, so it's not, everything is drastically simplified, but hopefully it will raise some interesting questions. So I think the first thing that really strikes me as interesting when thinking about risk and reward in the context of healthcare, and I'm going to be talking solely from the individual, the patient's perspective, comparing it to things on the financial and insurance side, is on the financial and insurance side, the option of a safe strategy always exists. So most of the risk-taking that's done is done from a base position of safety. I'm in treasury bonds, and I'm bored with the low returns and predictable outcomes there. I'm going to punt a bit of money on the S&P, but if I feel like there's too much volatility for me, I can go back to the safety of my government securities. And the same with the way an insurance company is run. There's a big pool of capital, and they will put increments of it against rare events. This is against hurricanes, this is against earthquakes, this is against tsunamis. But in total, I have a diversified collection of risks, and I'm monitoring very carefully the aggregate risk that I'm taking, and it's never above 0.5% over a 1-year time frame. Aging is, because of aging, the management of personal risk as it relates to how you decide to address your health is totally different, because as you get older, the safe option just disappears. And so I think what to me is very interesting about this is that it really changes the rational perspective on things which intuitively feel like they're dangerous. They may actually be rational when the alternative is natural aging. So this is a picture of a revolver. So I'm going to make a Russian roulette analogy. So assuming the revolver has six cylinders in the chamber and one has a bullet in it, if you engage in the game of Russian roulette, your mortality rate from doing that is 1 in 6. This is the mortality rate currently in the US for a 91-year-old. So if you're a general population US male, age 91, the chance that you will survive 12 months is about 1 in 6. And then this little this little dot down at age 50 is the point at which the annual mortality rate is 1 in 200. So this is the level of risk that an insurance firm will go up to as a maximum. So the regime of risk that's being discussed is totally different from what we usually see in finance and insurance. The converse of this Russian roulette example is if you decide to play Russian roulette, what's the chance that you survive it? 5 out of 6. And that has an analogy in human survival too. At birth, if a US male is born and mortality rates never improved from today forward, they just stayed at what they were, the chance of surviving Russian roulette is the same as the chance of a newly born making it to age 62. And so again, if a friend of yours comes up to you and says, I just engaged in a game of Russian roulette for the thrill of it, and I survived, it would sound amazing, and you would think they were crazy, but a 62-year-old is someone who has danced against the same odds and come out the other side. So to keep this as personal and narcissistic as possible, when you integrate, as is the purpose of conferences, when you integrate the various things that can impact your longevity, and I am focusing solely really on the endpoint of death, just to make things simple. The uncertainty is very, very wide. So this is 100,000 simulations of my life using the RMS longevity model, and this is just a histogram of the various outcomes. And what's striking about this, again, relative to financial applications that we will look at, is the width of this distribution and the number of decades which the space of possibilities is actually covering. So life is intrinsically a very, very risky business. And when I look at this, I feel afraid, if I'm honest with you. I just don't know what's going to happen. It's very difficult to plan against a distribution that's that wide. I have to finance my life up to some point on this x-axis, and there's a lot of things that can happen. So the baseline risk for a person is a very important consideration, I believe, when you're considering what types of therapy or lifestyle modification might be suitable for them, because what makes sense to one person might look like a desperate action to another person. So the classic example is someone who's trapped in a burning building might jump out of the window, even if they're a few stories up, which would be an irrational thing to do if in the absence of a fire. But for them, although it might shorten their life, there's also a good chance that it will extend their life. So as much as there's a really large challenge to understand the risk profile of different emerging technologies, because they always emerge in conjunction with an absence of any data on how dangerous they really are. The other side of the equation is understanding the baseline risk characteristics of different individuals. And this is something which is the topic of myriad epidemiological studies. And it's also something that the insurance industry has been collecting empirical data on for a while now. So again, there is precedent for fairly detailed models of human survival in the insurance sector, which take as inputs the disease states of the individual and the treatments that they're benefiting from. So this is a schematic of enhanced annuities. It's been a growth market in the UK, and up until recently, a quarter of new annuities were issued based on step one, filling out a very detailed questionnaire for the person who's trying to purchase a pension. So the idea is that the more unhealthy you are, the worse your survival looks to the insurance firm, the more money they will give you per annum for the same lump sum that you give to them. So the game is to pretend to be unhealthy when you apply for an enhanced annuity. This is the common mistake that you see. People are so proud of their health, they go to fill out the enhanced annuities questionnaire, and they're all showing off about all the various amazing things they have, and their biometrics are all under control. The net result is that they end up with a much smaller recurring sum that gets paid to them for the same lump sum paid up front. So, final section of this presentation is looking at a world where we assume that we can understand the risk associated with therapy X, Y, or Z, and our only job is to decide, now that we know the risks, do we want to take the risk? And this individual decision is the essence of sensible regulation, which is designed to make sure that people follow the optimum outcomes. And it's quite, I find it's quite a mind-bending thing to try to get a sort of coherent and generic grip on. So I'm going to give 3 examples of therapies and what they do to the survival curve of a person who might take them. And these should illustrate some points about the fact that there's probably a lot of individual taste around what type of risk and reward makes sense in combination. So this one is the easy no-brainer that definitely should be sailing through regulatory approval. A therapy that, once given to a 60-year-old, reduces the mortality hazard by 25%. So this is the annual risk of death at each age. The orange line is the US general population as of now, or as of 2011. And the gray line is the risk profile for a patient who takes this treatment, which just suppresses their mortality hazard by 25% at every age for the rest of their life. This pushes out their survival curve and increases their life expectancy from 82 to 84.5, so 2.5 years of life gained. Other than that, qualitatively, the shape of when they might die hasn't really changed that much. So there's nothing controversial at all about this therapy. In fact, at every single point, the taker of the therapy is at a better position than he would have been if he hadn't taken it. Globally, the mortality rates have been reduced at all of the ages. So that's the easy one. Now here's a second example. A therapy which temporarily reduces mortality to 0, but there's a cumulative effect that after taking it for a certain amount of time, it will lead to certain death. And for the financially minded among you, you'll see why I'm making up these pathological examples. It's to introduce the idea of risk tolerance and draw the analogy to that from finance. So this is what the mortality rates look like for this second therapy: 0 for 2 decades for the 60-year-old, and then 100%. The result of which is that the age at death for the person taking this is totally known. And so any upside or any downside is removed. And here is the histogram of the probability of dying at the various ages. I don't know, and there is probably literature on this, but I'm not aware of it. I don't know what is generally considered to be preferable by members of the general population. Would you rather die exactly on your life expectancy and know that that was going to happen? Or would you rather have the same life expectancy but have a spread of outcomes? I don't know. I don't think it's probably not that well substantiated what the risk appetite of the typical person is with respect to their own survival. And in fact, just out of pure curiosity, who here would prefer the gray risk profile versus the dispersed one? And who would prefer the dispersed orange profile? Okay, well that answers my question. the reason I prefer the grey one is because I actually see trouble at both ends of the orange one. Obviously, I don't want to die next year or the year after, so on the left-hand side, I'm not that happy. Conversely, I don't really want to get up to 100, 105 either, because I'm pretty sure if I do, I'll outlive my income, and I'll be destitute, and also I'll probably be terribly decrepit. It's very hard to plan against the orange one, and obviously I'm not trying to change anyone's mind about this, and it's all a fantasy anyway. But so in finance, when people think about their investment portfolios, they overwhelmingly prefer the grey one, and so this is a fundamental difference, at least in this room, in terms of people's appetite for risk. So a lot of the things which are true with the way people manage their money may not be true with the way people would like to manage their residual lifespan, which is a bit of a surprise to me. Okay, here's another one. This is the kill or cure therapy, which when you take it, you run a significant risk that your body reacts badly to it and you're done for. If that doesn't happen, you make it through the first year, then it reduces your hazard for all remaining ages by half. So this is one which elevates your mortality in the short term, but if you make it, it's the jumping out the burning building situation. If you make it, then you're in a much better situation. So here is a picture of the mortality profile. You can see at the very left-hand side, the gray curve, which is the curve for the person taking the treatment, is spiked up to a 5% mortality rate. So 1 in 20 people who take this treatment will just on the spot die. If they do not, then their mortality hazard will be much better going forwards. So you can see the survival curves. When you look at it like this, the gray line looks preferable. It instinctively feels greatly preferable to the orange line. And I think it's interesting to note that if you're focused on what will happen to me when I put this pill in my mouth or whatever it is, that's a very natural thing to focus on. But a rational analysis, if you want to call it that, is more represented by this more holistic picture of what will happen to your survival curve, would sort of clearly tip you in the favor of taking the treatment. But we're just talking about general population mortality. So the idea of going up to a 60-year-old and saying, take this thing, it might kill you with a 1 in 20 is popularly considered to be ludicrous. And I'm not suggesting I'm an exception to that. But again, trying to apply quantitative risk models to individual health and survival is a difficult area because whatever you might say comes out of a rational model, you can't ignore the political reaction and the gut instincts that people have. And in particular, the whole field of behavioral economics has shown that we're much more sensitive to losses than we are to gains. And this is also true in finance. If you make $2,000 on your portfolio, it feels quite good. But if you lose it, it feels a lot worse than it felt good when you made it. So we have this asymmetric response to the ups and the downs, which also complicates politically what's motivating a regulator because they're ultimately trying to keep people happy. So anyway, the result of this kill or cure is that the life expectancy goes up by 5 years. So at least under the life expectancy as a measure, this is a rational thing for a 60-year-old member of the general population of the US to undertake. So how possibly, it's because the survival curve, and I'm just talking about mortality as an endpoint, so this is as simple as it can be. Even in this very simplified example, and this is assuming you have all this information about what the hell will actually happen to a survival curve, and you can always come up with a model for that with the appropriate level of risk in it. It's still pretty complicated because the outcome is a vector of numbers. It's an arbitrarily shaped distribution that can take any form. It's still fairly complicated to decide what is good for an individual, and I'm sure it's different for different people. Here's a a crassly simple toy model of just for the illustrative value of how you could approach this problem. This is almost laughably simple, but the idea here is that, yes, you want your mean residual life to be higher. That's roughly a desirable quality. But you also care about what you do to the dispersion of your residual life. So most people here expressed a risk-philic attitude. Most of you said that I would prefer to have dispersion in my remaining lifespan. So I find that quite strange, but maybe I'm the strange one. So you people would say, I want that beta to be a positive number, and actually the more risk, the better. I don't want you to take away risk, because if you tell me when I'm going to die, it makes it too real, and I just don't want to accept it. So there's lots of interesting behavioral economics type things that go into this. Anyway, here's an example using this very simple formula of a quantitative policy for deciding whether or not a treatment strategy is worth adopting. So here I'm going to set beta equals -1. That means that I'm risk phobic, I'm averse to risk to some degree. If I made beta -infinity, I would have anything with any risk in it at all would be thrown out the window. Although your baseline situation, unfortunately, is pretty risky. So here is the baseline situation, assuming today's mortality rates are frozen and don't improve over time. You're a 60-year-old and you're a male. So, excuse me for that. So 82 is the mean time at which you will die. It's 22 years hence. And the standard deviation around that is 10. And so our objective function here, which is, or the utility, which subtracts the standard deviation from the mean, is 72. And we want to maximize this number. And in particular, if the strategy increases that number, then we're going to say, yes, it's a go, we'll adopt that one. So the global mortality reduction is the no-brainer. The mean goes up to 84. The standard deviation stays the same. The objective function went up, so yes, that one is approved. This one, where we know for a fact what the age at which we'll die, also approved, because the risk went away, and that gave us some payoff in the utility function. The kill or cure, with the 5% chance of the kill, increases the mean by 5 years, so we're up to 87 as a life expectancy, starting at age 60. It also does increase the standard deviation. It adds some risk. You've spiked the chance that a tail event will happen, so you've increased the dispersion of the outcomes. But with this parameter as it is, that risk penalty doesn't override the gain of 5 years in life expectancy, so it's an accept too. If it had been 20% chance of dying on taking the drug, it would have been a reject under this function. And you can calibrate this any which way. Here's a risk-neutral one. You set beta as 0, and the accept and rejects come out a little bit differently. And so none of my opinions are in here, but this is just an example of the type of quantitative measures that are used to govern the operations of financial institutions as applied to a very simple health endpoint. And already it feels like a minefield, just going through these pretty simple examples. It just feels wrong somehow. And so I think this is a very interesting topic for theoretical discussion, but a little bit scary as something that would actually be implemented. But hopefully that's thought-provoking. And my summary is that the world is full of misconceptions about risk and misalignment between rational measurement of risk and emotional response to risk. We all know that if a natural disaster kills N people and a terrorist attack kills N over 100 people, we're much more focused on the terrorist attack. The agent of the risk seems to make us think it's more risky than it is. And so there's myriad examples there, and they definitely apply in spades to David's panel topic that he's setting up here. So that's my slightly autistic perspective from the risk modeling side.
Thank you for a great talk, Chris. It's incredibly thought-provoking, and it's made me realise how skewed my risk perception may or may not be. Now, with an equally thought-provoking talk with case studies, quite whether it's as thought-provoking we'll find out, it's Brock Reeve.
Thank you, and it reminds me actually, years ago a friend of mine told me never appear on a panel with people with English accents because they always sound smarter and they know what they're talking about. Nonetheless, I'm taking on risk, personal risk. Unquantifiable, and who knows if I'll survive. Just a quick comment by way of background. The Harvard Stem Cell Institute is a research organization at Harvard that spans all the schools of the university and all the Harvard-affiliated hospitals with a goal of engaging from basic research with a stem cell perspective and bring it all the way into the clinic. And I'll give you an example of some of our work and some questions that it raises in terms of benefits, risks, and how do we value them both as individuals and as society. First of all, as a reminder, we talked about aging populations. Here's some US data. Why should we care about aging and the role that stem cells can play in this? And we can all find ourselves on one of these curves at some point or another, whatever our view of how we want to deal with it. And it's not just us. This is true around the world, and it's even more important in other societies, such as Japan is the example that everyone always points to. But you can see that this is really a worldwide phenomenon and one that's going to be a larger and larger phenomenon as we go forward. So what role do stem cells play in this? One, as you know, that part of the discussion around aging, and some of these sort of have been mentioned over the last couple of days, is we see the sort of repair and sort of process of maintaining homeostasis, of maintaining the condition that we like to think we're always in but never are, declines over time, and it shows up in a whole variety of diseases. And so the question is, how do we sort of think about that individually as well as systemically? So I'll give you one example, and by the way, there'll be a more detailed talk tomorrow on the skeletal muscle repair. One of the things that has been known for a while is that skeletal muscle regeneration declines with age. All of us feel that. We can't, run, jump, et cetera, with the same alacrity we could years ago. And this graph just shows you the difference between a young and an old mouse in terms of the number on the left and the size of regenerating muscle fibers post-injury. So the gray bars show you that an aged mouse repairs less well because it has fewer and smaller regenerating muscle fibers. Doesn't mean you should jump out a window, though. And so one of the projects we've engaged with, and this started with work at Stanford years ago and Harvard and elsewhere, is using parabiosis experiments where you join the circulatory systems of a young and old mouse together and say, wait a minute, sort of what actually happens here, and why is this true? And by joining circulatory systems, yes, it does mean you join the circulatory systems and hope the mice are nice to each other. And so what you see here in the gray bars that at the bottom right in both graphs say aged het, that's aged heterochronic pairs. The heterochronic mean both the young and the old joined together. And that the heterochronic pairs do a lot better than just the old pairs, the isochronic pairs, and almost as well as the young pairs in terms of, in this case, the test is rejuvenation of the satellite muscle cells, those cells that repair the skeletal muscle. And this has been known for a while that when you injure the skeletal muscle in these parabiotic pairs, that the old mouse in the heterochronic repairs almost as well as the young mouse. I don't know if there's a pointer here. No, that doesn't work. Anyway, if you look at the example on the lower right, the young-old pair, you see that the old mouse has been injured, And if you just look at the number of red cells, you see that the repair is almost as good as the young mice themselves, but the old pairs do not repair in injury. And then what we've done subsequently is saying, well, that's interesting. And what causes that? So there's stuff in the young blood, the young serum that's circulating that obviously, and this was referred to earlier in one of the questions, that leads to that repair and regeneration process. So we looked and we found a handful of circulating proteins and found one in particular, GDF11, that's a member of the TGF-beta family, and found that giving that protein alone could have almost the same level of effect. So for example, you see in the bar chart the fact of where the dark gray bars show the age control mouse, the age mouse without the GDF11. And when you give the GDF11 supplementation, you've shifted that curve to the same curve that the young mice had, if you remember that bar chart from a couple of slides ago. And you see down below that the number of cells and the size of the fibers is much better versus the old alone. And then you can see, well, they not only look better, but they actually do better. So here, this was a test where you basically put them on a treadmill and see how long they can hang on. Until they, exhaustion may be a little strong, but until they stop of their own accord, and you see that you actually get much better functional improvement. And that you can actually then look at the structure of the muscle cells, and it actually looks a lot better, et cetera. And so we took that work and said, okay, if that works in skeletal muscle, can that work in other organs of the body? And so we said, what happens in the heart? And this was a joint project with Rich Lee at the Brigham and Amy Wagers at the Joslin and Harvard. And one of the problems that the issue we worried about that is because age-related, as you see here, diastolic dysfunction of the heart is a major cause of heart failure. And at the moment, we have no therapies for it. So, and as you see on the cross-section on the left, the diastolic heart is, because it's gotten bigger, it's actually gotten weaker as a muscle, and it's gotten stiffer, and it basically is a bad thing. And there's basically no treatment for it. So we did the very same experiment, did the parabiosis, and if you look in the middle, the heterochronic pair, after they've been joined for 4 weeks, the old mouse, the cross-section of the old mouse's heart looks like the cross-section of the young mouse heart. So it's restoring that in phenotype. So, well, that's interesting. Sort of why is that true? And here it shows that it has, if you look at the pictures of the cells in the upper left, it shows that it's actually having an effect on the cell size, the cardiomyocytes that are making up the heart. So it's having an effect at the cellular level. And, whoops. And then we basically did the very same thing. So this was the parabiosis experiment, and then we did the same thing with GDF11. But before I get to that, I'll also mention, well, we also said, wait a minute, if it's, then we looked at, any other organ system, in this case, looked at the brain. And here, you see that, in the parabiosed animals, that in the heterochronic pair, that the old mouse had increased neurogenesis in the hippocampus in the brain compared to the old mouse itself, and increased blood flow. So we think the neurogenesis was driven, in fact, by the increased blood flow. So does that mean you want to get hooked up to a younger person all the time? You got to keep changing people as part of the problem. So what this did is so that if you look at, in this case, both muscle, heart, and brain, that in both parabiosis and in application of GDF11, the circulating protein, you could, in fact, restore the state to a younger phenotype and have more nerve cells, better heart function and shape, and better skeletal muscle function. Now, why this happens, what the mechanism of action is, we don't know. But we do know that it's a human protein, and we do know that in other studies that show that higher levels of this protein would correlate with better health outcomes over time. As a result of this work, sort of people got excited about it in the general press, and if you see, the various comments, And on the lower right, you see people say, well, could young blood be the fountain of youth? And so the press sort of got excited about it, but say, wait a minute, maybe we don't really want to be turning into vampires yet. So hold that thought, and I'll come back to that in a minute. And let me take a slight detour and sort of talk about a recent discussion about the pricing of Sovaldi, which is Gilead's product for hepatitis C. Why am I talking about this? That's because there's a lot of discussion around Gilead pricing this product for $84,000 for a 12-week course of treatment. It can cure hepatitis C. Some people say, great. Otherwise, it's costing the healthcare system anywhere between $350,000 and $500,000 to deal with these people with hepatitis C. This has a 90+% cure rate. So from a risk perspective, I don't know how Chris would think about it, but most people say, well, that's a good bet to take. On the other hand, people say, wait a minute, that can have a huge impact on the healthcare cost system. And in this article, this was from the Wall Street Journal last month, this article was talking about how the European regulators are looking at this very different and say, what's the cost and value to society? Yes, it may cure, but can we actually afford to do that or not? So in the US right now, the price is $1,000 a day and $84,000 total. And if you can read the fine print down at the bottom, it said Gilead had just agreed that in Egypt, the same course of treatment would cost $900. And say, wait a minute. $900 versus $84,000 for the very same product, very same results. How are we viewing these sorts of treatments from a society's perspective is something we need to think about if we can get to the point where, can we cure diastolic heart failure? Can we, keep our muscles, functioning the way that they used to? Can we restore our brain function? Can we cure hepatitis? And right now, this article, the European countries are all talking about, do we take different points of view? Do we all agree? Do we say, wait a minute, how do we think about this? Let's talk about Alzheimer's for a second, because one of the ideas behind this neurogenesis application of GDF11, could it be a useful drug for Alzheimer's? Alzheimer's, according to the Alzheimer's Association, has 5 million, and I think these data have been mentioned in passing over the last couple of days, 5 million people right now in the US, annual cost 200 billion plus. That's direct cost, Medicare, Medicaid, et cetera. 500,000 deaths per annum. Uncounted costs are 15.5 estimated by the Alzheimer's Association, 15.5 million caregivers, providing 220 billion in unpaid care, and themselves, because of both health and sort of psychological issues, causing about 9+ billion in direct health care costs. And that 5 million people with Alzheimer's is expected to grow to 16 million, more than triple by 2050. So right now, if you believe these numbers, we're paying 450 billion a year in costs related to Alzheimer's. So if something like GDF11 works and can have a major impact, how do we price it? How do we make it accessible? Can we say that we can avoid all these downstream costs, right? Can I cure diastolic heart failure? Can I help push out the onset of Alzheimer's? One of the discussions yesterday in the Alzheimer's neurosection was talking about these stages of disease, and once you get beyond stage 1, you're going to progress down the next few stages. Can we push that out? To whom do we make it available? And at what stages of the disease? And as part of that, are we bending the healthcare cost curve, which is one of the favorite phases when people are talking about insurance reform? Or are we just pushing the curve out, or are we actually increasing it? And at what point do we actually know that? Similarly, for the heart and muscle applications of GDF11, and again, let's assume it works, and let's assume it does what we think it might be able to do. Do we approve the drug only for specific indications? I.e., is it only for muscle wasting, for cachexia, or sarcopenia, or for diastolic heart failure? And let alone approve it, do we pay for it only for specific indications? Or can anyone who has a sore muscle say, okay, I want some of that, or do I want to use it prophylactically? I gave an example of hip fractures. A lot of elderly people fall over and get hip fractures and have to get operated on, and their bones break because they have osteoporosis, but one of the reasons they fall is because of muscle weakness. So if you can prevent the muscle weakness, can you avoid hip fractures in the elderly? Well, if that's true, how do we get compensated for this? Because you're taking a drug, do I get paid by the drug, or do I get paid by the value of avoiding all those downstream healthcare costs? And if an elderly person is laid up with a hip fracture, you have all the health, the indirect costs as well. What happens on the other side? That's not only illness, but wellness, or people want to be extra well. What about the bodybuilders, the athletes? EPO is a great product, but some of the Tour de France riders sort of got carried away with it. And the Tour de France said, okay, EPO isn't legit. You can't increase your red blood cell count. But on the other hand, if you can increase muscle strength here, what's okay in private situations, what's okay for individuals, what's okay within sports or other sort of regulated environments, and what's okay in the healthcare system, and how do we value and price those? So I think the science is getting to the point where we're raising those questions. I don't have any answers. You notice I've only asked the questions. But I think we need to start asking those questions as we think about the risks and the benefits, because the science is pushing us to that pretty soon and pretty quickly. And as you see with that, with the hepatitis C example, we're already dealing with that today, and we're only going to, and we haven't been ready for the answers on that. And we're only going to have more and more of those questions facing us going forward, thanks to some of the new technologies that are really, that are coming out of the lab now and coming into the market really, really quickly. Thanks.
Thank you, Brock, for a provocative talk. And I think that was fascinating. It's a different type of case study. He's really looking at the technologically driven change as opposed to the provocative numbers game Chris offered. Now it's my pleasure to introduce Anant Johani from Better Value Healthcare, who hopefully can demystify some of the pricing issues which have been raised by all three panelists so far. And whilst Brock may ask for questions, Anant has the answers.
Well, no. We try to give payers the answers. So the work that we do at Better Value Healthcare focuses primarily on working with the payers, the guys who are actually going to be paying for these innovations. So we don't deal with any of the regulatory things or anything like that. We work downstream with the payers who are going to decide whether or not they're going to pay for something that has gone through the regulators and is now out on the market. And we call these hellish decisions that they need to make. Because what has been sort of described so far is individual technological innovations, whether they be drugs or diagnostics or even nowadays with the IT solutions that are coming to the fore. How does a payer actually decide what to fund, right? I mean, the hepatitis C drug that Gilead has come up with, that won't be the only option for patients with hepatitis C, right? And so there's also a preventative component for that. How can you do something from a public health perspective to prevent people from getting hepatitis C in the first place? And how much should you allocate for that? So the thing that we focus on when we're working with payers is value. And so value is a term that's sort of commonly thrown around for all sorts of stuff. And at Better Value Healthcare, we're big fans of Wittgenstein. So we believe that language creates your reality. So we like to define the terms that we're working with. So if I can ask you just to spend 2 minutes and speak to your neighbor and come up with a definition, how do you define value? So 2 minutes, and then we'll have a sort of a quick feedback session. If there's not a neighbor close to you, just find the person closest. 2 minutes. That's 2 minutes. Okay, 2 minutes. 2 minutes, must stop talking. Can we get some feedback from the audience? Anybody want to volunteer? Yeah? Utility, okay. Others? Quality? What you're willing to give up. Something like opportunity cost? Betterment or to the receiver. You were saying? Relative metric. Any others? Anything else? Yeah? Yeah, benefits to whom? Yeah. Yeah. Others? Okay, so values is quite tricky, isn't it? Because then you can also talk about, if you use the word values in the plural, that's like personal values. I value equality. I value diversity. I value right to speech. These sorts of values. So for us, we see value divided into thirds. We talk about triple value. So we divide it into 3 different types of value. There's technical value, allocative value, and personal value. Technical value, allocative value, personal value. So technical value, we use Michael Porter's definition, who's a professor at Harvard Business School, and he defines technical value in healthcare as patient outcomes over cost. Patient outcomes over cost. Allocative value we define as how do you allocate resources for the maximal benefit of the population. So that's a population level metric. And then you talk about personal value. How do you ensure that the healthcare you're delivering is meeting the needs and expectations of the patients that you're serving? So technical, allocative, and personal. So these are the 3 things that we talk about when we're helping payers decide what to fund and what not to fund. And in America, I mean, we don't like talking about rationing of care, but the people that are paying for healthcare are rationing. Medicare and Medicaid, they're deciding what to pay and what not to pay for. You know, Brock came up with this example of Gilead. Should we be paying $84,000 for a treatment for hepatitis C, or should we be using that money, that $84,000, and invest it in, say, exercise regimens to make sure that people who have diabetes can prevent diabetes from exacerbating and becoming even worse? How do you decide where to allocate those resources? When you have a finite budget, you have to make these decisions, and these are what we would call hellish decisions. And there are ethical dimensions to this that are not fun to deal with. And we don't have the answers. I wish we did, but you know, this is what we help payers decide and work with. And then there's a patient dimension, the personal value. How do you ensure that you're actually meeting the needs and expectations of your patients? So I'll give you an example. There was an orthopedic surgeon in Oxford, and a woman came to him, and she said, I need a knee replacement because I have trouble bending my knees. And I have pain in my knees. Sorry, I have pain in my knees. So the orthopedic surgeon, you know, did the test, and he said, okay, yeah, fine, you know, you qualify for the surgery. And then he just happened to be talking to the patient about why she actually wanted the surgery. Was there any other means that she could use to, you know, overcome that pain? And she said, well, she wanted the surgery because she was having trouble bending her knees while she was gardening. And she wanted to get the surgery so that she would be able to garden pain-free for 2 or 3 hours at a time. And then the surgeon said, well, do you realize that if I replace your knees, you're not going to be able to bend at all? You know, not the way that you were able to bend before you had your knee replacement. And she said, no, sorry, I didn't realize that. And she got up and left. And then she took, you know, analgesics and things like this. So although the surgery would have been of high quality, I think somebody mentioned quality, it wouldn't have been of high value to the patient because it's not meeting the patient's needs and expectations. So it could have been high quality, i.e. there are no complications after the surgery, the surgeon did a fantastic job, et cetera, recovery was fantastic, but it would have been a low value to the patient. So how do we ensure that we're actually delivering high value to the patients with any of these innovations that are coming out onto the market? Are we actually meeting the needs and expectations of patients? If we're going to allocate resources to that innovation, where does that money have to be disinvested from so that we can actually invest in that innovation? And will that actually lead to better outcomes for the population? And then how do you actually look at the outcomes versus cost? Something like the Gilead drug, for instance. At $84,000, the outcomes would be X. And in the US, it would be X over 84,000. In Egypt, the outcomes would be X over 900. So the same drug would be of much higher value in Egypt than it would in the US. So these are the sorts of things that we work on. We've started working with innovators as well to help them to start thinking about and framing their innovation in the context of what payers have to be thinking about. Because at the end of the day, they can go through the regulators and they can get their drug out into the market, but if the payers are not going to be willing to pay for it, then it's just going to sit on a shelf somewhere. So that's the stuff that we work on. I'll leave it to Dave to facilitate the group discussion.